Good morning, everyone, and welcome to the Garvan Institute of Medical Research. It is such a joy and an honour to welcome so many members of our Garvan family in person and in the Zoom room and via Facebook Live to celebrate Garvan's 60th anniversary. We are honoured to be on this land today, and I'm personally humbled to pay my respects to the traditional owners of the lands on which we meet, the Gadigal people of the Eora Nation. We pay our respects to elders past, present and emerging, and welcome any Aboriginal or Torres Strait Islanders with us, either in person or joining us virtually today. It's also a privilege to acknowledge the people that came before us, the Sisters of Charity and the St Vincent's Hospital Sydney. And here today we stand on the St Vincent's Hospital Sydney Health and Innovation Precinct. A little bit of housekeeping. For those of you in the room, in the unlikely event of an emergency, please follow my instructions at all times. The exits are via the front of the auditorium or the rear of the auditorium, and the preferred exit is via Burton Street, with the exception of individuals who need lift access. For anyone who were to require lift access, please congregate at the front of the auditorium. I've been thinking a lot about today because I've had the privilege of working at the Garvan Institute for more than 10 years. And I appreciate that's nothing compared to 60. But I've been thinking a lot about what motivates people to become involved with the Garvan Institute of Medical Research. It does come back to that vision of the Sisters of Charity, these incredible women who came up with this idea of not only treating disease at the hospital, but finding out the underlying cause of disease, which is the only way that we can improve diagnoses, treatments, prevention, and ultimately cure. So today we stand on the shoulders of all of the people who've come before us. And I think that what's most unique about Garvan, in addition to the quality and excellence of our medical research and our brilliantly talented scientists, is our Garvan community, our Garvan family, all of you and many, many more who are personally, intellectually, psychologically motivated to support medical research. There is not a doubt in my mind that the success that we've had to date is because of the support of the Garvan family. And so to all of you, those of you with us in the Zoom room and those of you who can't be with us today but might watch this at a later time, we thank you for your transformational support that has enabled the Garvan Institute of Medical Research to genuinely be a national and global leader in the areas of science and health that we are dedicated to investigating and making demonstrable impacts on human health. It's now my great pleasure to introduce Professor Peter Croucher, Garvan's Interim Executive Director, long-standing Deputy Director, a pioneer in the field of skeletal disease, bone biology, osteoporosis, but also cancers of the bone, which, as we all know, are the last and final outcome for cancer. And currently there are no cures. Peter has been doing an incredible job steering Garvan over the past 9 months, for which we were most grateful. And I'm delighted to welcome him now to provide the official welcome and opening of today's public seminar, 60 Years of Discovery. Thank you, Peter.
Thanks, Margaret. Okay, well, thank you very much, Mara-Jean. And I'd like to add my welcome to everyone here. It's wonderful to see so many people in the room. It's also awesome that we can now have people join us via Zoom and through other means as well. So yeah, it's pretty special to see so many people in the environment and have you all visit us here at Garvan. We're here to celebrate 60 years of Garvan and the research discoveries and the impact that the Institute has had for medical research. And Most of you will be aware from Mara-Jean's initial opening words that the Institute really was founded out of a department within the hospital 60 years ago today. And it was really that vision of the Sisters of Charity and their passion for coming up with ways of treating disease, preventing disease, that they set about trying to raise money to create an institute. And fortunately, a major donor came forward, Mrs. Helen Mills, and she gave a donation of £100,000, which at that time in the 60s was an enormous donation, and that allowed Garvan to be created. And in recognition of that particular donation, it was agreed that the Garvan Institute will be named after her father, which was Patrick James Garvan. And as we now know, that's the inception of Garvan as we know it today. Now, our first director of Garvan was Les Lazarus, and he was an endocrinologist, and he spent his time in the UK training in London before he returned to Sydney and to the Institute, and he was really the founding director of Garvan. And he was really an individual that really pioneered endocrinology in Australia, and brought that into the Institute and had a real focus around diseases like diabetes and bone disease, something that I feel particularly passionately about, and also seeing the introduction of research into cancer. And sadly, Les passed away just before Christmas, and we're looking at ways of celebrating his contribution and his role within Garvan at points during the course of this year. What I'm really pleased about, though, today is that we have three former directors that you're going to hear from, John Shine, John Maddock, and Chris Goodnow. And you're going to hear from them about their various experiences of leading Garvan at different stages. And individually, they are absolute legends in the field, pioneers of the field, and real visionaries in terms of thinking about how to take medical research forward. And at each stage in Garvan, we've been privileged to have these individuals help step us through the next phases of Garvan's research. You're also going to hear from Warren and Sue about different elements of what Garvan has done, both in the past, but also in a particular area of science that Sue's leading at the moment that probably has a really major role to play in the future as we go forward. But I think it's fair to say that Garvan has grown enormously, obviously, since those early days, probably from a small number of individuals in the department. And when I was talking to Ted, he was talking about 20, 25 individuals, to an organization that now has nearly 700 researchers, scientists, professional staff that help support medical research. So we have grown enormously as an organization, and matured as an organization now, and we're highly regarded as a medical research institute, not just across Australia, but we're known internationally for the research that we do. And I'll always remember that I knew of Garvan as a PhD student when I was in the UK 30 odd years ago, and when I was writing up my PhD in Cardiff and then in Cambridge, I was looking in the literature and I could find all these publications from the Garvan that described the wonderful work going on in the osteoporosis field at the time. And I think since I've been at Garvan, I've seen many more of those discoveries shared with the community. And I think it's fair to say that Garvan is very much on the international, global picture for medical research. As far as going forward is concerned, we've spent time over the last couple of years looking at where Garvan is really strong and where we're really contributing. And that is built upon many of the discoveries, many of the ideas that you'll hear about today that have come forward from John, Chris, and John as well. And we're really building upon that to take us into the future. And as Mara-Jean has said, I'm currently in the interim role, and we're going to be joined by Benjamin Kyle in a couple of months' time. And Benjamin is going to be the next director of Garvan. And there's going to be amazing opportunities created for Benjamin to help take us forward into the future. And you'll have plenty of opportunity to engage with Benjamin in the coming months. So we'll create those opportunities, and there'll be plenty of more times when you can come together to actually meet various members of Garvan. So today is very much about celebrating the successes of Garvan over the last 60 years, but also an opportunity to think a bit more about the future, how we shape that future and what Garvan can look like. And I'm super excited to be part of that at the moment and hope we'll be part of that going forward. I think there are enormous opportunities and it's going to be an exciting time going forward. But today is about celebrating what Garvan has done and achieved over recent years. And I'm going to stop there and hand back to Mara Jean and let her introduce the next speaker. So over to you, Mara Jean.
Speaker really needs no introduction. Dr. Warren Kidson, forgive me everybody, a clinician scientist who played a major role in Garvan's discovery, which is currently celebrating its 50th birthday. This is a really important discovery in endocrinology with particular relevance for individuals living with type 1 diabetes, which is a disease that predominantly affects children, young people and young adults, but has a profound impact not only on the kind of health and well-being of individuals, but importantly on the quality of life. And Garvan really has a big legacy in this space in terms of driving and clinically implementing some of the biggest transformations in type 1 diabetes. Dr. Warren Kidson, please get up here.
Well, good morning, Garvan friends. I'd like to thank Peter Croucher for inviting me to present today because this work we did 50 years ago has really made a big difference to diabetes. Now, it doesn't look like my... No. Here we are. So, 50 years ago, we developed a new treatment for a life-threatening high blood sugar state called diabetic ketoacidosis. This work was done by John Casey, who is watching this presentation from Brisbane, Ted Cragen, who's in the audience. Can you stand up, please, Ted? There he is over there. Ted has been associated with Garvan for 55 years. And of course, our director, Les Lazarus, who passed away last December, has been mentioned. Les is one of the finest brains I've ever had the pleasure of meeting, and I'd like to dedicate my presentation to the memory of Les. Now, type 1 diabetes is caused by a complete loss of secretion of the hormone insulin from the pancreas gland at the back of the tummy. What does insulin do? Well, basically, insulin controls energy usage throughout the body. It gets tissues to suck up glucose for either for energy use or for storage for later energy use as something like something called glycogen, which is like starch in the plant world. So plants store energy, glucose in starch, in grains, etc. We store glycogen and animals store glycogen in their livers and their muscles. Insulin stops protein from breaking down, and very importantly, it stops fat from breaking down. Now, type 1 diabetes is a complete loss of insulin and leads to this condition called diabetic ketoacidosis, which I've abbreviated as DKA. So a complete lack of insulin causes a breakdown of our glycogen stores in liver and muscles, and a breakdown of protein as well to glucose. And it blocks glucose entry back into the cells. As a consequence, the level of glucose in the blood rises, it goes out through the kidney tubules, dragging water with it, causing dehydration. And just as importantly, we have uncontrolled fat breakdown in our body. Fat's broken down to something called ketones, which by nature are acidic. So the body becomes acidic, and that impairs functioning of the heart and brain. Now, prior to the discovery of insulin 100 years ago and the introduction of insulin therapy 100 years ago, type 1 diabetes was a death sentence. The person with type 1 diabetes would die within 1 or 2 months of this condition called diabetic ketoacidosis. But insulin changed all that. One teaching was that in ketoacidosis, insulin didn't work very well. There was a resistance to the efficacy of insulin, and you had to give large doses, 50 to 100 units under the skin or into the muscles every 1 to 4 hours. You and I produce about 60, 80, perhaps 90 units of insulin per day, per 24 hours. Fluid was given into the veins for rehydration. Recovery was slow over 1, 2, 3, 4 days after many hundreds of units of insulin. And the death rate was around 10 to 13%. Not a nice prospect. What did we do to change all this? We took 11 patients with either very high blood sugars or ketoacidosis and gave them some fluids into their vein to restore their circulation. And then we started trickling in this tiny, tiny dose of insulin. Let's see if this... No, we can't get this. Not to worry. So we would start by giving these people 1¼ units of insulin per hour for an hour or two. Then we would up that to 2½ units of insulin per hour for a couple of hours. And finally up to 5 units of insulin. Now, the amazing thing was that we found that the rate of fall in blood glucose was maximum at 2½ units per hour. Doubling the dose of insulin didn't get a faster response. So we established that 2½ units per hour was the optimal dose. And interestingly, that dose gave the patients the same level of insulin in their blood as you and I would have after a meal. So we virtually disproved that insulin was not inhibited by this condition. There was no insulin resistance. By 6 hours, mostly the glucose levels were back to normal, and we'd only given them 15 units of insulin, not hundreds of units of insulin. Now, what about the impact of this research? Our work became the subject of editorials in most major medical journals throughout the Western world. Professor Philip Felly from Yale summarized it in the New England Journal of Medicine a few months after our publication. And he pointed out the insulin levels in the blood were the same as you and I would get after a meal, disproving the existence of insulin resistance. And he correctly said that insulin infusion was a simple, safe form of therapy that could be administered even in tiny hospitals without much in the way of backup. It didn't have to be in a major hospital with lots of resources. Now, this research also went off in a different direction. Our research established the baseline for insulin therapy in the artificial pancreas. And that was done by Ted Cragen, who's here today, Leslie Campbell and Don Chisholm, all the Garvan, and was done elsewhere in the world. The impact of this research has recently been the subject of a review by the NHMRC in Canberra. But we had to go a bit further than that because parents of children with diabetes told me that they didn't want their kids to go back to hospital yet again. So I thought about it and decided that one of the most common causes of diabetic ketoacidosis was anxiety. Anxiety in a person who had given an insulin injection but wasn't taking food in because they were vomiting. Parents shared their anxiety, often doctors shared that anxiety, and they would stop the insulin. So we had to get them not to stop the insulin without the sugar going low. So I suggested a plan where they would sip small, frequent mouthfuls of sugary drinks, lemonade or Coca-Cola, not diet lemonade. So that would overcome the vomiting problem reasonably well. The other problem was that of hypoglycemic coma, low blood sugar coma, and that the sugar could be got back up to levels where the brain could function again with the injection of a pancreatic hormone called glucagon. And finally, if a person was drifting into high blood sugar states, early diabetic ketoacidosis, we found that by giving an injection of a small dose of insulin every hour into the muscle had much the same effect as low-dose insulin infusion. So the person with diabetes or their parents could get the blood glucose level down without needing to go to hospital. They'd usually talk to their diabetes specialist by telephone, but they didn't need to go to hospital and the problem was resolved. So 50 years after our work, low-dose insulin remains the treatment of choice for diabetic ketoacidosis around the world. Mortality has dropped from around 10 to 13% to less than 1%. And we are very happy, we're very satisfied with the outcome of this research. Thank you.
Thank you, Warren. Truly transformational and impactful research done right here with your leadership. Well done. Audience, you'll never guess who's coming next. Let's go back to the professor who needs no introduction, Professor John Shine, Executive Director of Garvan for more than 2 decades, and fortunately for us, somebody who is still conducting research right here in the building on level 7. John is famous for many, many things. He discovered the Shine-Delgado gene sequence and went on to be the first leading pioneer in the initial world-breaking sequencing or cloning rather of human insulin and human hormones, which have had a huge impact across a whole range of diseases. These days, John is transforming polycystic kidney disease, which is a really dreadful familial disease, in conjunction with colleagues at St Vincent's Hospital. And in his spare time, he quite likes the races and the horses. Come on up, John.
Thank you, Mary Jane. Thank you very much. It's obviously a great pleasure to be here and to see a few old familiar faces too. Thank you so much for your support of the Garvan, and I won't go through all of the reasons why medical research is so important, and the Garvan's doing its best, and I think we're doing a great job. And it's a real privilege for me to be here with 3 other ex-directors, 2 other ex-directors, 3 of us all together. I must remind you guys, you remember that saying about rooster today, feather duster tomorrow? Yeah. So it's good to be here with the other feather dusters. And I've only got 10 minutes, so I'm going to have to be quick. And I'm really just going to try to, in that 10 minutes, all I can really do is touch on the sort of underlying thrust of what was driving Garvan's research over the past few decades in many ways. You'll get lots of specifics, as we've heard from Warren already. Fantastic work, and I'm sure John and Chris and Sue will give you some other specifics. This was just a sign we had up at the front of the Garvan foyer, of course, for many years, which to my mind summarises what the Garvan was really all about. And that is imagination based on knowledge is the key to discovery. I mean, humanity is rich in imagination, always has been. But our knowledge base in the last decade or 2 has just been exploding, growing exponentially. So if you take that together, it really is an age of discovery. And I'm so proud that Garvan's been able to participate so strongly in that discovery. What I'm going to talk about very briefly today really is just, as I said, some of the underlying things that are driving some of this research. And it's been all about increasing knowledge of genes and the human genome and what that means for the genetic basis of disease. And then I'm going to briefly touch on how that affects our understanding of multifactorial disorders like cancer and heart disease and osteoporosis, all the ones that challenge modern society, and that interaction between genes and the environment. So let's briefly just touch, I'm sure you all know, but I'd love to go over it again, the human genome. The human genome, as you know, is there's 3.2 billion of these GATCs, 4 simple chemicals arranged like beads on a string. And if anyone in the audience would care to dedicate their body to science, and we homogenized you and extracted your DNA or your genome, it would reach to the moon and back about 8,000 times. So there's an enormous amount of information there. If you typed out the 3.2 billion GATCs of your genome in the font size of a Sydney telephone directory, any individual would fill up 500 200-page directories. If you typed it out in a 12-point font, it would reach from Sydney to Brisbane and back twice. So it's a lot of information that is there, an amazing amount of information. And certainly having been around a little while now, I can assure you, as Warren will tell you, 60 years ago, this was considered a dream to have access to that sort of knowledge base. We are, of course, about 99.9% identical in our 3.2 billion GATCs. Everyone in this audience is about 99.9% identical. But that does leave the other 0.1%, there's about over 3,000 little differences. You might notice on that slide, if you can see the little red G there, that could be, if this was a small part of my genome, that little red bit could be a mutation or a change, where it's G instead of, say, an A, which accounts for the difference, a very subtle difference, between myself and Tom Cruise. And this is really what tells us the differences. So as I said, it's 3.2 by 3.2 billion of these base pairs long. That accounts for, now we know, about 30,000 genes scattered throughout that. If you do the math, genes about a few thousand GATCs long, if you do the maths, you end up saying, well, that's only about 1.5% of the genome encodes for the proteins, the hormones, the enzymes, the structural proteins that do all the work in our body. That leaves another 90%, which was unaccounted for. For quite a while, that was thought of as sort of junk DNA, because about half of that was highly repetitive DNA sequences, which were thought to have been left behind in the genome by past virus infections, which had evolved along in a degenerate form with our genome. And the rest was just considered junk. I'm sure John Maddock, who's a real expert in this area, is going to tell you about how that whole part of the genome now in recent years, we know has much more important functions as well. And it's opening up a whole other area. The United Nations, being the United Nations, in 2000, when Bill Clinton and David Blair on opposite sides of the Atlantic made the big announcement about, we've got the first draft of the human genome. Politically, they wanted to score lots of points. The United Nations had to jump in, of course, very quickly, because they always do that sort of thing, and had to have their definition or their declaration on the human genome. And it's actually quite spot on. The human genome underlines the fundamental unity of all members of the human family, because we are 99.9% identical. But it also is a recognition of their inherent diversity and dignity. So in a symbolic sense, it is the heritage of humanity. And it's that heritage of humanity that Garvan's been able to harness as we take our research forward. It's often called the Book of Life. I think this is my first general slide about it, so I won't bore you too much more. But it's actually 4 books. It's a history book because it contains a detailed history of human evolution and the migration of humanity around the planet. It's a complete construction manual and parts list because it contains all of the instructions needed to develop a complete human from a single fertilized egg cell. It's a great medical textbook because it has within that GATC 3.2 billion a description of every gene that has any effect on every disease we have. It's also a who's who in the world because it shows the differences that make us unique between individuals. So it's a very powerful database of knowledge. I could tell you about lots of different things that we've already learned. One just vaguely interesting thing that happened to come up was that men were, the mutation rate in sperm is about 6.5 times that it is in the egg. So men create inheritable mutations about 6.4 to 5 times greater than that of women. So that allows me to immediately say with great confidence, men are the primary driver of evolutionary innovation. My female colleagues, I can see Sue, she can't wait to see something. Sue will point out that no, it just means that men are the primary source of genetic errors. So that's science. One data set, two totally different interpretations. But we've learned a lot. We're learning a lot from the human genome. I'm going the wrong way. Oops. I've already mentioned that we're 99.9% identical. We're 98% identical to chimpanzee DNA, for instance. So clearly the differences between species are not just in the numbers of genes or numbers of proteins, but they're in how those proteins are regulated and interact and in a complex organism. As I mentioned earlier, although we're incredibly identical, there is many millions of differences between everyone in this audience. They're not scattered randomly through the genome. They're often concentrated in particular areas. And that, of course, is the basis of forensic medicine. Because each of us, although we're 99% identical, each of us is absolutely unique in certain sequences. It really, what's it done for our research though? Not just for at Garvan, but around the world. It's really transformed how we do medical research. It used to be very hypothesis-based. And that is, if I take prostate cancer as an example, a researcher may notice that particular prostate cancers seem to make a lot of a certain growth factor. So their hypothesis would be production of this growth factor is driving the cell to become cancerous. And they'd test it out experimentally. Now with the whole human genome there's a lot more emphasis these days on discovery-driven research. And that is you make no a priori assumptions. You just look at the whole genome of a prostate cancer cell, the whole genome of a normal prostate cell, and you say, what's different? And you'll see various gene mutations that are different, and that will give you an idea to go and test then whether what mutation is driving that particular cancer in that individual. And that points you to a new way to look and treat. I mean, hypothesis is very critical still, but it's added to with this discovery-driven research. In a very short period of time, over the last 10 years, we've now got databases with hundreds of thousands of complete genome sequences from lots of different populations, from lots of normal, so-called normal populations, people without any particular disease, from genome sequences from groups of patients with particular diseases, and from many different ethnic groups. When you compare all this data, you can rapidly identify particular changes or mutations which are driving specific diseases. And that's adding enormously to our ability to move from this discovery into figuring out what to do about it. It would not be possible, of course, without the rapid development over the past couple of decades of these sequencing technologies and the bioinformatic analysis of those sequences. I just should point out, of course, when they made this big announcement of the first draft of the human genome, which was only about 83% complete, it wasn't even the whole thing, it was just a draft in 2000. It had taken researchers 15 years and about $3 billion to come up with that one single draft, that one single genome sequence. A decade later, with the development of technologies, it took about 2 months and cost about $20,000 to get a similar sequence. Today, really, as we all know, especially at the Garvan and similar research institutes around the world, you can do a genome sequence in a couple of days and it costs probably about a few $100s. Maybe a few more $100 to do the analysis of the genome, but the technology has developed incredibly. And this is just a rough graph here from the Human Genome Institute in the US. It just shows a comparison with the cost per doing DNA sequencing per genome with Moore's law. I'm sure most of you know Moore's law is the equivalent in the computing field. It's basically if I get it right, the number of transistors that you can put into a microchip doubles every couple of years and the price comes down every couple of years. And that was considered fantastic, that exponential sort of development of the technology. If you look in the green curve here, of course, you can see with the human genome sequencing, the cost, which took 3 billion to get to this first point, and then it was 100 million per genome, very quickly has come down over the past couple of decades to now that couple of $100, which makes it a more feasible to be a fairly standard diagnostic sort of test. How do we apply that, especially at the Garvan? Really to understand mostly multifactorial disorders, diseases which have as their cause both genetic and environmental factors. And identification of the particular genes that are involved in those disorders will give rise to new approaches to prevention and treatment. The sorts of things we're talking about are there, every one of the major disorders that challenges modern society. I mean, I suppose examples are simple. If you pick something relatively straightforward, like heart disease, we all know being overweight and perhaps smoking and certainly drinking too much and no exercise, you're at more risk of having a heart attack. That's the environmental risk factors. On the other hand, we've all heard of the thin 40-year-old, really healthy, probably vegetarian, healthy 40-year-old drops dead at 40 years of age, jogging through the park. And that's the genetic factors. He or she, unfortunately, in life's genetic lottery, had some mutations in genes which made them susceptible to heart disease. The other end of the spectrum, we all know about elderly gentleman, 90 years old, overweight, goes to McDonald's every day for a good feed, drops dead on his 90th birthday, in bed, smoking a cigar with his third wife. I mean, that's the genetic and environmental interplay. Most of us are somewhere in between. But that gene and multiple genes and the products of multiple genes and their interaction with environmental factors makes an enormous difference in our risk factor for certain diseases. So if we can identify the specific genes involved, then of course you then have a target to develop specific therapeutics. In the meantime, you can help with prevention. And if I follow on with that cardiac disease example, even before we have particular treatments based on genes, if there's 10 50-year-old men go into a doctor's office and he has a chat to them all and he says, look guys, you're all overweight, most of you smoke, you drink too much, you don't exercise. 6 of you 10 boys will have a heart attack by the time you're 60, unless you do something about those environmental factors. What do? They all look around and say, I wonder who that poor guy's going to be. You know? But if you can take a blood sample from each of them and you can say, you, and you will have a heart attack if you don't do something about your environmental challenges because you have a genetic mutation that makes you susceptible to having a heart attack. So you can practice prevention with that knowledge even before you've got the actual sort of answers. 2 quick examples. I've probably run out of time already. I'll try to be real quick. Cancer and obesity. Cancer, for example, we know there's many ways to look at cancer, but one way is to look at cancer. We may hear more about this from Sue. If you think of cancer, the uncontrolled growth of a cell leading to cancer, you can think of it a bit like there's 2 classes of genes involved. Oncogenes, which make the cell multiply, and tumor suppressor genes, which tell the cell not to multiply. Those 2 classes of genes act like sort of accelerators and brakes on a car. So if you're a normal car or a normal growth pattern of a cell, and you get, as the cell's slowly dividing, and you get a mutation by chance in one of these accelerator genes, the car starts to go a little more quickly. But you've still got your foot on the brake, so it doesn't move much. If you then, by chance, get another mutation in the brake gene, the car starts to take off more. Then another mutation, and of course, the faster the car goes, or the faster the cell divides, the more chance of another mutation. So you get this cascade of mutations in accelerators and brake-type genes, which drive the development of a cancer through these various phases until you get a really terrible sort of drug and radiation-resistant cancer at the end. So if we can identify, as is happening with all of this research, particular genes in any individual's cancer, which are driving this cancer in that sense, it gives you, again, a source of a target to develop a way to treat the cancer. And often you can use a drug, if you know the gene driving your breast cancer is the same one mutation that's driving a lung cancer, you could maybe try a drug that is working in breast cancer on a lung cancer in a fairly rapid way without having to go through lots of other stuff. Sorry, another last quick one. Obesity. We've got a sort of circuit in the body that determines our set point of obesity. We all know, you can be overweight and you can diet and do everything right again. You can lose a bit of weight, but as soon as you stop, boom, you're back to where you were. And that's because we heard a bit of this from Warren. It relates to Warren's insulin stuff. When you eat something, you absorb the food, you metabolize it, and you lay down fat in the fat cell, the adipocyte down the bottom there. The more fat in that fat cell, the more this gene, the OB gene, the obesity gene, the more that gene is turned on and makes the hormone leptin, from the Greek leptus, meaning thin. The more of that hormone that is made, it goes up to the brain, interacts with its receptor. In the brain, that triggers the release of all sorts of other neurochemicals, which affect your appetite, your preference for certain sorts of food, and your metabolism. And that then leads to you eating more, and around you go. So if your obesity can be shown to be due to, say, a mutation in the leptin receptor, then a drug targeted at that particular receptor is likely to be most effective in treating your obesity rather than someone else who has a different sort of mutation for their obesity. So it's all about this progress in prevention and treatment of multifactorial disorders. It really involves this understanding of the gene-environment interaction and allowing us to then apply that in a personalized way to an individual with their individual thing. Prediction, prevention, individualization. I can just end then on imagination based on knowledge is the key to discovery. Thank you. Your turn, John.
I think it's fair to say that Professor John Shine is the Tom Cruise of the Garvan Institute of Medical Research, which is why I did allow him to go 8 minutes over time. But I'm going to have to be particularly strict with our next speakers, so apologies in advance. So please join me, John.
Right.
As Professor Shine has mentioned, John, among many other success stories and many other discoveries, is really the godfather for the whole paradigm-shifting discovery that junk DNA, also called junk DNA, is in fact not junk. John, we are thrilled to have you here. You really do only have 10 minutes.
Thank you, Mara-Jean, and thank you all for coming today. I'm not going to talk about junk DNA, although I'll slip something in at the end since it's a topic in terms of its roles in complex disorders. But actually, John's done me a great favour because he's done a great primer on genomics. So that'll save a little bit of time for me. Because what John pioneered, and he's too modest to say, is that he was the person who cloned the gene for insulin, one of them, and other important hormones. And the period from the mid-1970s to the 1990s was really the age of gene cloning. And the advantage, and he did that in California before he came here, but the advantage for the community is that people who suffer diabetes now have genuine human insulin as a treatment, rather than the pig insulin that used to be purified from the abattoirs, which caused all sorts of secondary problems. So we now have a cloned human gene, and the protein is produced in bacterial yeast for medical purposes. It's a huge advance, and it extends to growth hormone and many other important hormones for treating human diseases. So I came in 2012, and it was just on the cusp of the change really from gene cloning and analysis to genome analysis at scale, for reasons that John has mentioned. And this was the period of the advent of genomics, and I want to talk about how we introduced that at Garvan and the impact it had particularly in cancer, because I know Chris will talk about it in immunology and Sue will talk about it in terms of epigenetics. And this is really the true situation. And we're in the early to middle stages of this total transformation of healthcare, where we use personal genetic information to inform relative risk and the correct treatments. So, as John has pointed out, but I'll move quickly, we each have about 3 million differences between us that underlies our individual physical and psychological differences, and the basis of the diseases that all of us in one capacity or other will suffer through our lifetimes. And understanding this basis is critical for diagnosing and selecting appropriate treatments and preventative strategies, as John has outlined. So John's mentioned this. This is the, on the left, the two covers of the journal, Journal Science and Nature, announcing the first draft of a human genome sequence in 2001. And as he said, the costs came down as fast as Moore's law until about 2007, when there was a step change in the technology, and they dropped hyper exponentially down, because that's a log scale on the left, to about 2012, 2013, when they leveled off, and which is when I came. And we were very lucky at that point, because the technology then took another step change down to $1,000, which made it feasible to apply for research at scale and in the clinic at scale. And we were one of the first three in the world to acquire this technology, thanks to a very generous donation from John and Jill Kinghorn, the Kinghorn Foundation, people who gave the foundation donation under John Shine's leadership for the Kinghorn Cancer Centre. And so we started to apply this. John and Jill were also very generous, and this is the importance of the support for the Institute, in not only supplying the money for the $10 million to buy this equipment, but also the money to staff it and to develop the analytics so that scientists and clinicians could use it. The computing, the software, and the establishment of a team of people who could run these machines internally and help people with their bioinformatic analyses. So we also needed, of course, funds to support the application of this research in cancer. And I was very fortunate to travel to Los Angeles at that time and convince the International Alliance Foundation to provide funding for a pilot project for kids with cancer, which was then followed up locally by David Thomas, who was then director of the Kinghorn Cancer Centre, Marie Zizek here, and Michelle Haber at the Children's Cancer Institute, with whom we had a fantastic partnership. And in fact, I'm going to give you one case study here that brings this to life, but a report of the 247 initial participants in this program, and you might have noticed on the news a day or so ago that this is now going to be rolled out much more comprehensively, that children who received a targeted therapy based on genomic information, of those, 1/3 went into partial or complete remission. So I want to tell you about Ellie's story, because she was one of the early examples of this. And we did this in conjunction with the Children's Cancer Institute. And that's Ellie there. She was diagnosed at 11 months of age with a sarcoma in her chest. Now, sarcoma is a cancer of connective tissue. And this, she was dying because this tumor in her chest was too large to surgically remove without killing her. She was on oxygen, and all hope was nearly lost. So we were able to sequence her tumor, and Mark Cowley, who's then here and now works at CCI, identified the cancer as being caused by a rare translocation in a particular gene called NTRK. And as luck would have it, a company in Seattle had just developed a new drug called larotrectinib, which blocked the action of this mutant protein. So we're able to get it across here for Ellie, and within 4 weeks, her cancer has shrunk to the point where she was off life support and could breathe on her own. 6 weeks later, she was home, and she was later declared disease-free. Amazing. Now, this is the power of that level of information that John talked about. Now, down the hall from Ellie, another little girl, this case hasn't been reported, was suffering a glioblastoma, which is a lethal brain cancer. We sequenced her tumour. She had the same translocation. She was treated with the same drug, and I'm advised that she has now been declared disease-free as well. Now, and the figures are now showing that 1/3 to half of all children will have improved survival, if not lifetime remission, with this information. So this is the contribution that Garvan has made in conjunction with partners to improving the lives of kids with cancer and generally with other people with cancer. In fact, on the basis of this and other cases, the clinical trial was open in Australia for all children whose cancer has the same translocation, but you can't tell from looking at it because the location of the cancer and the look of the cancer depend on which cell the mutation occurs in. In fact, before I got here and under John's watch, Andrew Biancan, working with Sean Grimmond at my old institute in Queensland, was part of a big international team that showed exactly what John was talking about, that cancer is a very heterogeneous disease at the genetic level, and all of these coloured bars represent a different mutation. You can see if you look at pancreatic cancer on the right there, it's caused by a whole range of different mutations, many of which are treatable by targeted drugs, but you don't know which drug to use until you get the information. So based on that, David Thomas and Mandy Ballinger have built on that. They had a series of trials called MOST and other nice names to test the feasibility of doing sequencing followed by targeted treatment. And they engaged the drug companies to provide the drugs for free for this trial. The trials were so successful in principle that the federal government contributed $65 million in a giant partnership, $190 million led by Garvan, with commercial partners who are supplying drugs for free, or the Children's Cancer Institute, the National Computing Infrastructure in Canberra, and then other partners around the country, both hospitals and health organisations, to do a giant trial across Australia to look at the value, both in terms of medical and health outcomes, but also health economic outcomes, of instituting cancer genome sequencing as the standard of care in Australia. So I think the contribution that Garvan has made and is making to the future of cancer treatment is just extraordinary, and will usher in a totally new treatment away from just getting blasted with radiation and actually having your cancer treated based on an intimate knowledge of what's really causing it. So we also, during that period, established the Garvan Weitzman Centre for Cellular Genomics. Chris had a major role in this. This allows us to look at fine detailed individual cells, because every cell in the brain is different, every cell in the cancer is different, in the immune system, to get fine scale information for research about the molecular events that underpin both normal development and disease processes. And this is now headed by Joseph Powell, who arrived just before I left in 2018. So we are moving into a new era, and I think of all the institutes in Australia, Garvan's been the one that's led this in this country, towards precision and personalised medicine, a revolution in medical research, healthcare and health economics. So data from patients will be generated from genome sequencing, from clinical records, but also I think smart sensor data, and it's what we're already thinking about, talking to Apple and patients about downloading stuff from, because that's actually more accurate on the whole than clinical data. It goes into a big database, which allows us to diagnose and prognose conditions more accurately and treat with correct treatments. It also, of course, feeds directly into research, this huge database, including the use of artificial intelligence, and then ultimately to transforming the health of the population and the costs of healthcare delivery. This will be the solution to the healthcare cost prices of the future. And that's my sign to get off, but I'd just like to thank you for coming and for supporting this fabulous institute, and to continue to do so, because what Garvan does and what institutes like Garvan do is create futures that often are ahead of the curve from conventional and orthodox peer review systems. have tremendously gifted people who really think ahead of the curve, and they can only do that with the support of the community. So thank you.
Thank you so much, Professor Matic. And only 2 minutes over. I'm very proud of you. Thank you so much. Our next guest is Professor Susan Clark. Sue's CV is more than 3 pages long, but the highlights are that Sue's genuinely a globally recognised and celebrated pioneer in epigenetics and epigenomics. She is one of few Australian scientists to have many, many accolades and many awards and prizes over more than 2 decades here at the Garvan Institute. She's the current NHMRC Senior Principal Research Fellow and Head of Garvan's Epigenetics Lab. Sue, thank you so much. Coming up.
Thank you. Well, thank you, Mara Jane, for the introduction, and thank you all for being here today to be able to hear about our work at the Garvan. And so today I'm going to share our research beyond the genome that we've heard about from the 2 Johns, and delve into our world of the epigenome, which is the information above our DNA sequence, and explore what our understanding of the epigenome is and its implication that it has in the understanding of cancer as well as the treatment of cancer. So as we've been hearing about DNA, the structure was discovered now 70 years ago by Watson and Crick. And the genome was then sequenced 50 years later to discover that there was this 3 billion letter code. But this left us with a dilemma, because if each cell has the same DNA, the same genome, how can each cell type be different? And so this is the dilemma that I was really interested in addressing. And the answer is because each cell type has a different epigenome. So what is the epigenome? Now, we just heard about the genome, and that's the complete sequence of our DNA, the 3 billion letters. But the DNA isn't naked. It's actually surrounded by proteins within the cell. And this is one of the first pictures that I like to think of the epigenome here. It's an electron micrograph that was taken in 1973 by a husband and wife team, Donald and Ada Ollens. And here you can see DNA as beads on the string. And the DNA is wrapped around these beads, this protein. And the function of that is to divide the genome into different portions, a bit like the grammar is to DNA. So if I take this analogy a little bit further, and we heard from John, the genome is a collection of our 3 billion letters. But to read those letters without any grammar is really difficult. So if you just read these letters here, it will take you a while to work out what that says. But the epigenome provides the grammar to that DNA. And so now if I was to put punctuation here, I could read that as a woman without her man is nothing. Now, I don't like that. So therefore, if I was a different cell, I would change the grammar, not the sequence of the letters. But if I change the grammar now, it reads, a woman without her man is nothing. Now, this shows you, and it shows me, the power of the epigenome, the power of the grammar to change the meaning of our DNA sequences. And this is how each cell type that has the same genome can have a different meaning and become a different cell type. So I'm going to delve a little bit further into what I also mean about the epigenome. So we heard that in DNA, 3 billion letters, if you stretch it out, 2 meters of DNA. Now, if we zoom in to the DNA, it's part of our chromosomes, and our chromosomes live inside the nucleus here. And so to be able to fit those 2 meters of DNA into a tiny little nucleus inside the cell, it has to be organized in a really tight 3-dimensional structure. And part of the what the beads on the string does is it enables that DNA to be organized in this really tight structure, such that some parts of the genome are open and spread out on the string, whereas other parts are closed and protected, and so that DNA is silent. And what that does is it means that different parts of the genome are either active or silent in different cell types, which explains why a heart cell might be different to a muscle cell, which might be different to a brain cell, because they have different parts of the genome silenced or active, depending on how that DNA is organized inside the nucleus. Now, one of the other key punctuation tools that causes our genome to be changed is a chemical process called DNA methylation. And this is a process that I've been studying now for over 30 years. So DNA can be chemically modified, and this chemical modification, called DNA methylation, occurs by putting this methyl group on the C base of our DNA when it is next to a G base. So this CG base is susceptible to methylation, and there's 28 million of those combinations in our 3 billion letters, showing that there's a great possibility for methylation across the DNA. Now, when DNA is sequenced, and we heard about the Human Genome Sequencing Project, all you could read were the 4-base code, and the methylation status of the DNA was lost. And so we didn't know which sites were methylated and which sites weren't. And so back in the early 1990s, my lab and I developed a new sequencing protocol that allowed us to read not just the 4 bases, but to read whether that cytosine base was methylated or not. And that has totally revolutionized how we can interpret these differences in cell type determination. So we went on to use this methylation sequencing to see how the epigenome changes in development. And we were the first to show back in the before the 2000s that the DNA methylation pattern actually is removed during fertilization. So it removes the methylation at the C sites, and then methylation is reset during development. So each individual person has its own epigenome. And later on, we and many others have now shown that DNA methylation patterns actually erode as we age. And so our cells become less healthy and our methylation has not been maintained. And so this provides a foundation for why diseases can happen. And so one of the diseases that we've primarily been looking at is the development of cancer. And so we've applied methylation sequencing to cancers. And again, back before the 2000s, before I arrived at the Garvan, we were the first to show that tumor suppressor genes that John Shine talked about, retinoblastoma, is not only mutated, but can also be methylated in retinoblastoma tumors. And the P16 tumor suppressor gene also can be methylated. And so this was really revolutionized the field of cancer, to think it's not just about gene mutations. So in the 2000s, it was then clear that at a single gene level, cancer-related genes can be both mutated and methylated. But we were interested then to find out, well, this is at specific genes. What happens genome-wide to our epigenome in cancer? So this is when I decided to join the Garvan in 2004, my methyl group. We were working at the University of Sydney, and the Garvan really gave us the possibility to be able to explore new technologies, more sophisticated technologies, to answer these questions genome-wide. So I established our epigenetics group here, and over the last 18 years, we've published over 110 papers about how the epigenome changes in cancer. And our groups divided our projects into 3 main pillars. The first is understanding the mechanism of how this works. And so we've created the first epigenome maps for breast and prostate, normal and cancer cells. And what I mean by that, in its analogy like mining, is that if we delve deep into the beads on the string, we've compiled the layers of the genetic information, DNA sequencing. Then we've put another layer of what the methylome looks like, another layer of the beads of the string structure to find out which regions of our genome are active and which genes or which regions are silenced. And more recently, we've been able, I'm not sure why that hasn't come up. More recently, we've been able to develop a new technology that allows us to look at the three-dimensional organization of the DNA inside the cell. So this is a huge amount of information above our genome sequence that we can now understand how it changes in cancer. And from these maps, we've been able to discover for the first time that whole regions of the cancer epigenome are changed, whereas before it was only thought to be single genes. And moreover, these large-scale changes change the three-dimensional architecture of the DNA and how it's packaged inside the nucleus. And the importance of this is because it gives us now a mechanism of how genomic changes occur and how different programs in the cancer cell become resistant to drugs. The second field of work that we've been pursuing over more than a decade is to use this knowledge from these maps to develop new biomarkers for cancer. And in particular, we are developing now blood-based tests to be able to look for early detection of cancer, to be able to assess whether the cancer is high risk or low risk, and to assess whether these cancers are responding to therapy, and to be able to use a molecular test to see when the cancers start becoming resistant. So these are all based on being able to profiling the methylation mistakes that occur in both breast and prostate cancer. And this just shows many of the publications where we're reporting these findings. And the third area of work that's more recent is to look at new therapies. So epigenetic therapy is a therapy that removes the DNA methylation. And we're exploring whether low-dose epigenetic therapy for the treatment of breast and prostate hormone-resistant cancers actually would work. And so we've been doing a large amount of preclinical work to show that if we remove these methylation marks from the cancer, we can restore the three-dimensional architecture back to what it was in a normal cell and restore the gene expression. And that is resulting in these cells dying. So this is a work in process and one that we've now been able to initiate a phase 1 trial at St. Vincent's Hospital across the road. So we've gone from knowing that cancer was a disease of the DNA to now really establishing that cancer is a disease of both the genome and the epigenome. And the next steps, which is where the field is going, where our lab is going, and where the Garvan research is going, is to be able to exploit this knowledge to further develop these epigenetic biomarkers, because unlike genetic biomarkers, they can be used for multiple cancer types, and they can be used in addition to the specific genetic changes that we've heard about. And in particular, exploit epigenetic therapy as a new way of being able to treat cancers. But in the future, we still don't know many things. We still don't know the fundamental principles of what changes the epigenome in cancer. If we know that, then we could start looking at ways of preventing it, so to try and prevent cancer from happening in the first place. And the second really major question is, I only talked about cancer today, but we know that the epigenome can change in many diseases because the epigenome is critical to define a cell type. And when a disease occurs, the epigenome changes. And these are studies that are now being pursued by many groups in the Garvan, in immune disorders, neurological disorders, and in diabetes. So it's a really exciting future, one that I hope that you're inspired to see how Garvan is developing. And finally, I just want to acknowledge my team who worked incredibly hard over COVID. This is part of our Zoom meetings we used to have lots of fun at. And they're all very excited to pursue this work. Thank you.
Thank you, Professor Clark. How inspiring, everyone. It's just remarkably important work. Our next speaker is Professor Christopher Goodnow, globally renowned immunologist and proud recipient of the Bill and Patricia Ritchie Foundation Chair. Chris was Executive Director of Garvan from 2018 to 2022, prior to which he was Deputy Director from 2015 to 2018, following on from an illustrious career at ANU and Stanford. And we are so thrilled that Chris is retaining his research at Garvan with his HOPE research effort, and just delighted to have him speak with us today. Chris, come on up.
Thanks, Mara Jean. And boy, big boots to fill here following this crew of people. But I'll talk a little bit about immunology and try and bring some of the different things that you've heard together in a field that's captivated me ever since I was training originally to be a veterinarian, a lot like John Shine did before we both discovered medical research. So let me tell you, I want to talk about immunology firstly to tell you, how did Garvan get into immunology? And it started, as I understand, there'd always been there, but a bequest, a really generous bequest that allowed two things to happen. One was to recruit a brilliant team from a biotech company in Boston, Charles and Fabienne Mackay, and to recruit some of the best minds here in Sydney and bring them all together to build on all of the other work that you've heard about and take it into immune diseases. And that really has just gone from strength to strength. It's synergized with all of the other things that you've heard about. But I'll just talk about one thing, and that's antibodies. Now, antibodies you've heard a lot more about in the last couple of years with the pandemic than we ever heard before. They are a miracle of nature. They're a magic bullet that zeroes in on its target in a way that there is nothing else in nature like it. It binds with incredibly tight affinity, these magic bullets, but with incredible specificity. There is nothing else like it. And we know so much about both the way that they do this, and I'll show you a movie in a second, and how our bodies shape these antibodies to have this exquisite specificity has only been possible because of the tools of gene cloning and genome sequencing that you've just heard about. That really opened things up for me and many, many other people. Now, one of the critical things about these antibodies and their exquisite specificity is they've got to be directed to the right targets. So they need to see a virus and not see part of our own body. And that really is at the core of a whole lot of genetic machinery that goes into shaping the way that the antibodies fold and recognize their target. So in this movie, the antibody is gold, of course, and the target, which is in blue. And it's, in this case, a target that is very similar when it's a normal part of our body or something that you've been vaccinated against. And what we were able to do, and what's really summarized in this movie, is to learn exactly how the antibodies, through a series of checkpoints, are redeemed from the sin of binding to a normal part of our body, which could cause autoimmune disease, and mutate the fingers of the antibody so that they no longer bind self, but only bind with very high affinity to a foreign target. And that's at the core of this miracle of how antibodies work for so many different things. Now, with some viruses, they've actually learned how to exploit this specificity of antibodies to their advantage. And that's the problem that we're living with right now with the pandemic and coronaviruses, is that most of the antibodies that recognize the coronavirus recognize this one patch on the coronavirus, which is shown here. It's this region here with all these yellow surfaces. And so almost all of the antibodies that protect us from infection with SARS-CoV-2 either after a vaccination or after infection, the majority of them are targeted to this one spot on the virus. And when Omicron exploded on the scene back a year and a bit ago, it acquired all these mutations, all these yellow things that you can see there on the screen are changes in the virus target, so that almost none of the antibodies could bind any longer. And that's why we suddenly had another great big outbreak, even though we were all vaccinated. Now, why I'm focusing on this is because there are rare antibodies that get around this virus trick. And just last week, this paper appeared from a very large team of people at Garvan, at the Kirby Institute and others, led by Professor Daniel Christ, where he was able to isolate 3 antibodies from people that had recovered from infection that get around this trick. And so this one here that I'm showing, this green antibody, Garvan-5, recognizes this patch, but in a way that carefully moves its fingers around to avoid the bits that are changed. And what we're really hoping is that this antibody and 2 others that are described in this paper, that Daniel and his team will be able to take them forward as a cocktail for a new way to treat any coronavirus infection in the future. And in parallel, what we're doing is to work up a vaccine strategy to elicit more of these kinds of antibodies and less of the antibodies that the virus can quickly escape from. Now, one of the things I wanted to point out in showing this is you can see in each of the slides the large team of people that are involved in any one of these projects at Garvan. And that's really one of the other things that's changed, I think, over the last 60 years, from just, you know, the solo scientist with a Pasteur pipette and a couple of Petri dishes to really large teams of people working across very many different disciplines and technologies and different institutes and around the world. And the list of people on this paper is an example of that. And that's really, again, one of the joys of being at Garvan is it brings together all of those disciplines. It reaches out around the world to bring people together to solve the problems. Now, some people can't make antibodies. And back when I used to run the medical immunology course for the medical students at Stanford, we knew of a very small number of devastating diseases in children where they couldn't make antibodies or immune responses and were terribly vulnerable to infections. And the treatment for that is to give them antibodies exogenously from the company CSL, and I'll come back to that in a moment. But we had no tools back then to really figure out why most of these kids had these inborn errors of immunity. But what you can see in this graph from a paper that just came out recently is this extraordinary growth, thanks to genome sequencing around the world and global collaboration in the number of different diagnosed diseases, explanations for why these inborn errors of immunity arise. And you can see there are now over 450 as of last year. And what's really special about this is this global panel that gets together every year to review and collate and assemble the list for all clinicians around the world is chaired by Professor Stu Tengi, who's here at Garvan, and really spends half of his time talking with people around the world, other clinicians and immunologists, to make sure that we know exactly what the diagnosis is for any child that comes in with an unexplained problem with their immunity. Now, the thing that's even more special about antibodies, and only became possible with gene cloning and genetic engineering, is the ability to use them as drugs. And they're now actually one of the most fastest growing classes of new drugs in drug development. And there's a couple of dot points that Garvan's contributed in different ways to that side of things, and I just wanted to share them with you. So one was a long time ago when I was at Stanford and giving those lectures to the medical students. We discovered that the way that the immune system gets the right specificity to antibodies and avoids autoimmune diseases through a series of immune checkpoints. And I'll come back to that in a moment. One of the things that happened here at Garvan in 2001 is with the recruitment of the Mackays and the extraordinary team of researchers here, not only did they figure out some amazing things about how antibodies are controlled, but they also spun out a startup company to actually make antibodies as drugs. And that company was called G2 Therapies. And in 2008, it licensed an antibody to Novo Nordisk, one of the big pharmaceutical companies, that's being taken forward as a potential treatment for inflammatory disease or cancer. And there are many, many different antibodies around the world that are going that way. But here's another data point. And that's from 2011. John's trying to get me off. But if you're not impressed by science, you have to be impressed by share prices. And so John took, when he stepped down as head of the institute, took on the role of being chair of Commonwealth Serum Laboratory, as it was once known, CSL Limited, which produces the lion's share of polyclonal antibodies that are given to people with inborn errors of immunity and also for many neurological diseases. And during John's 10-year tenure as chair of CSL, the share price of CSL went up tenfold. So we're claiming all of that for Garvan. All right, and I wish I could claim it, but that little discovery of immune checkpoints back 35 years ago or so did turn into a completely new way to treat cancer with the development of immune checkpoint inhibitors that now is routinely used here in the Kinghorn Cancer Centre and anywhere else for melanoma, for lung cancer, for kidney cancer. There are now hundreds of thousands of people around the world who have been cured or bought 5 or 10 years extra life as a result of being able to take the brakes off the immune system by giving them antibodies as drugs that reactivate the immune response against cancer. And here at the Garvan and at many other places, including my own lab, we're working on ways to get that to work in more people and more kinds of cancer every day. Now, this is my last slide, Mara Jean. You've got 30 seconds. 30 seconds. All of that's great, but what really brought it home for me when I joined the Garvan was the ability to bring all this knowledge about antibodies together with all the technology about genomics that the two Johns have described, and to interact with all the hospitals around Sydney and actually see, is this actually really good for real people, really? And so within a few years of joining Garvan, that all came together, and the two examples that I wanted to share was Alan and Carla, who were both kids, one at Sydney Children's Hospital, one out at Westmead Children's Hospital, who had an inborn error of immunity, but despite having the best doctors you could have, they didn't know what it was. And they were not responding to the usual treatments. And so in Alan's case, he was in intensive care, about to die from a lack of platelets. In Carla's case, she couldn't walk due to the pain from having terrible arthritis as a very young child. But the genome sequence not only gave us an explanation for the inborn error, but it immediately said, hey, there's a genetically engineered antibody drug that these two kids should really respond perfectly to. They should be exceptional responders. And so we could work with the hospitals and the clinicians, give them the evidence they need to get the approval to do that. And in both of those cases, the outcome was a miracle. And the real privilege was to see them come into the Garvan Institute. Carla, completely bored with the science of immunology. She just wanted to know where were the cupcakes. And Alan terrorizing the lab staff with his lightsaber. But that's what it's all about. And I guess just to cap off the point for everyone, it all starts and it all happens because of all of you, the community, and your generosity, both with cash, which is crucial, but also with your support and encouragement. It's one of the reasons that all of the people at Garvan love to work here, is because you come in and you tell us what we're doing is important. Work harder, do it better. So thank you.
Thank you, Chris, and thank you everyone for joining us today. I will ask our audience, both online and in the room, to give a huge round of applause to our speakers, Warren, Professor Shine, Professor Matic, Professor Clark and Professor Goodnow. As per usual, we would value all your feedback, and you will receive a little survey to fill in following on from the seminar. You will also receive the opportunity to join us for the next seminar, which will be taking place on the International Women's Day, which I believe is Wednesday, the 8th of March. So if you'd like to come back in person or online to join us to hear from some of our incredible female scientists, in addition to Professor Clark, who's done a wonderful job today, we would absolutely love to see you there. As Chris has said, our Garvan community is precious. We are humbled by your support. We are inspired by your advocacy, and we look forward to another 60 years of huge success for Garvan's medical research and its impact on human health. Thank you so much, everyone.